This is Ritik, your biology subject matter expert from MEMONEET, and this week we are going to once again concentrate on this very important chapter, that is cell cycle and cell division. As most of you have asked me to do a revision video on this chapter, that's why I have taken this chapter. Okay. So basically, what do you mean by cell division? How cell cycle happens and all? See, coming to the very important point, even the sperms that we see and the ovum that is produced, they are cell, right? So you can see that the cell after like before fertilization and after fertilization, how gradually they multiply, and from very small size to how the size of the cell, that gets extended. Like cell starts dividing, multiplying, becoming its own specific function, playing its own specific role, everything. And finally, they start becoming what? A specific organelles, then an entire body, right? So this is how, there is some kind of transformation that the cell will go. Like after fertilization of the egg, what happens is you will be, going to see that two cell stage, then four cell stage, we have 8, 16, then the blastocyst. You know the zoology, like especially in the human reproduction, right? I've explained to you like the following stages. So this is how during the stage, the individual cells, they multiply. So very important concept that comes here is, okay, there is one individual cell here, and this individual cell is actually multiplying into two cell. Then to that, it is multiplying into four cell. Fine. So we know that there is a chromosome, and even there are other cell organelles and all, right? So how they have been equally distributed from one cell to their daughter cell, from parent cells to the daughter cell, how they have been equally distributed. This is very first question that comes in mind. And how DNA has been replicated. Like whatever the chromosomes we have, whether they're equally they're sharing or not, that also comes in mind. Like we have how many chromosomes? 46 chromosomes, where 44 are autosomes, where 2 are sex chromosomes. So is the 46 chromosome is going to transfer to the next cell immediately? So this all question comes in our mind. Yeah. So to tackle it, this chapter plays a very important role. To know exactly what is cell division and cell cycle, and what are the process and events that occurs in different phases of cell cycle, to know this chapter is very important. So cell division is a very important process in all living organisms. During the division of the cell, what happens? The DNA is going to replicate, and the cell growth also takes place. This is one point. And these process, that is, what do you say, the cell division, then the DNA replication, and the cell growth. These are the three major events that happens in cell. Hence, they all, they have to take place in a coordinated manner, that is, in a proper, specific manner. There has to be a coordination, a correct division, correct DNA replication, correct cell division. Okay. So, why? Because and formation of the progeny cells containing intact genome, a proper genome, a specific genome has to be present. If this doesn't happen, that is proper cell division doesn't happen, DNA replication will not happen, or irregular replication will happen, which leads to what? Mutants of cell, right? And specific non-functional cells. The sequence of events by which a cell duplicates its genome, synthesize the other constituents of the cell, and eventually it is going to divide into two daughter cells, is termed as cell cycle. So when where you are actually seeing eventually how the cell cycle is happening, where it is going to duplicate its genome, that is the chromosome, then synthesize the other constituents of the cell, that is like cell organelles, and eventually it gets divided into two daughter cells. Okay. So that is actually called as cell cycle. So cell growth. Uh, is a continuous process. That is, it doesn't stop at one specific time. Okay. So almost most of the cell growth, whatever you'll be seeing, is actually a continuous process. So cell growth in terms, I'm actually talking about the cytoplasmic increase. Okay. So then we have the DNA synthesis occurs only during one specific stage in the cell cycle. That is, we have specific cells cycle, right? Cell cycle stages, right? Prophase, metaphase, anaphase, telophase, and interphase, right? So even in interphase, we have what? We do have G1 phase, synthesis phase, G2 phase, right? So in one specific phase, like say synthesis phase, so the DNA synthesis app occurs. That is what they're trying to say. That is, DNA synthesis occurs only during the specific stage of the cell cycle. So, what is that specific stage? Let us know it. So, coming to the cell cycle, during the duration of the cell cycle can vary from organism to organism and also from what kind of cell and what kind of function the cell is going to perform. So, say like human cell is culture in culture, that is an artificially or basically human cells. It will take approximately 24 hours for cell division. Whereas if you consider yeast, it will take around 90 minutes cells, okay, will actually take 90 minutes to complete its cell cycle. So there are basically two phases of cell cycle. One is interphase and M phase, which is nothing but the mitosis phase. An interphase, what happens? The phase through which the resting phase is the time during which the cell is prepared for division by undergoing both the cell growth and DNA replication in an ordinary manner. So this is a phase where the cell actually comes up with the resting phase, and now it will eventually, after this phase, it will be ready to, grow to the a kind of cell cycle process. Eventually, meanwhile, in this phase, that is interface, in this interface, what happens? The cell will be preparing itself to for division, undergoing both cell growth will occur and DNA replication will also occur in this phase. Okay. So you will be seeing three kinds of further phases in the interface. Say like G1 phase, synthesis phase is nothing but S phase, and G2 phase. That is gap 2. Okay. So you can see the figure here, that is the interface figure, how the, the chromatin is present, how the chromatin are like, thread-like structure, and centromeres are two paired. Fine. We have plasma membrane, that is the outermost layer, and nucleus is also present here, right? So you are also seeing here like this is the cell cycle, and you are seeing G1, that is cap 1 phase, S is the synthesis phase, and G2 phase here. So what is the major role of these phases? G1 phase, okay, that is gap 1 phase. You can see here like how they are. So the G1 phase corresponds to the interval between the mitosis and initiation of the DNA replication. That is, so where is it? So this is the M phase, right? And this is what? The S phase. So in between M phase and S phase, what you are actually observing is G1 phase. So it is somewhat, it acts as a mediator between the M phase and S phase where the DNA replication will take place. So G1 phase corresponds to the interval between the mitosis, that is M phase, and initiation of the RNA replication, that is synthesis phase. Okay. So that is what G1 phase is. And during this G1 phase, what happens? The cell is metabolically active and continuously grow but does not replicate its DNA. That means we know here there is a DNA, and metabolically it is active, continuously it grows, but do not replicate its DNA. Okay, this is very important. Then coming to the yes phase. After G1 phase, we are going to find what? Yes phase. So in yes phase, which we normally call as synthesis phase, which is marked, which marks a period during which the DNA, replicate the synthesis and DNA replication takes place. Okay. So during this time, what happens? The amount of the DNA per cell gets doubled. If the initial amount of the DNA is denoted as 2C, say like we have 2C of DNA, then that is going to increase to 4C. That is, duplication will happen. The DNA will replicate, DNA will also synthesize. So more DNA, you are going to see the twice of what DNA it has. However, their chromosome number will be exactly the same. That is nothing but you are going to see the chromosome number will be same. Okay. That is, sorry, what do you see that? 2n, which is if the cell is diploid, or 2n number of chromosomes at G1 phase, that is 2 into, what do you say that? 23. Okay. So 23 pair of chromosomes you are going to see which are uh, haploid in nature. Then another two chromosomes are actually what? Uh, yes. So basically, they're the sex chromosomes. So basically, what happens is if there is 2C amount of chromosomes is 23, obviously what you're going to see, you're going to see the increased number of the DNA, but not chromosomes. So chromosomes initial count will be the 46 itself when it comes to the humans. Okay. So that's what they're trying to say here. So in animal cell during the S phase, DNA replication begins in the nucleus and the centrioles duplicates in the cytoplasm. Coming to the G2 phase, which is gap 2, during this G2 phase, the proteins are synthesized in preparation for the mitosis while cell grows what? Continuous. Uh, the next phase that we have is G0 phase. So G0 phase is also called as quiescent stage. So some cells, what happens? Not every cells eventually keep on dividing. Okay. So once they reach in adult animals, once they reach to the specific position, so they will start. They are going to stop their cell cycle process. So some cells in the adult animals do not appear to exhibit division. Say like heart cells and many other cells where the cell division only associated as needed to replace the cell that have been lost because of injury or the cell death. So if there is necessary, then only the cell division will happen. If not, no cell division is going to occur. So these cells that do not divide further exhibit what? G1 phase to enter into. From the G1 phase, they are going to enter into inactive stage, that is G0 stage, okay, of the cell cycle. Cells and the stage remains metabolically active but no longer prolifies unless they are called to do its task depending upon the requirements of the organisms. The next very major important phase is M phase, which is actually called as mitosis phase. So the M phase represents the phase where actual cell division that occurs, and the interphase represents the phase between the two successive M phase. Okay, that is between two division of cell, whatever this, like once the cell is ready to divide, right? So now, what happens? The cell, this is a parental cell, and it got divided into two cells, right? Two daughter cells. Now, this daughter cells will also reach as parental cells, and it has to divide, right? So, for division, whatever is required. So from the parental cells to two daughter cells, the cell divide, it gets dividing, right? So there, between that initial stage, it is called interphase. So it is significant to know that in the 24 hours average duration of the cell cycle of the human cell lasts only about 1 hour. That is, if it is a cell cycle, so only 1 hour you are going to see the cell, cell division properly occur. Where remaining will actually takes more. That is 95%. Okay. So yeah, only this much, only what do you say that 1 hour out of 24 hours, only 1 hour will be M phase, where remaining rest will be the uh, interphase. The M phase starts with the nuclear division corresponding to the separation of the daughter chromosomes that is actually called as karyokinesis. So your two important keynotes will come, one is karyokinesis and another one is cytokinesis. So I don't want you guys to, get confused in this specific terms. So karyokinesis is nothing but the separation of daughter chromosomes. So in cells, inside the cells, chromosomes are present. So there is a separation of daughter chromosomes will occurs at one specific stage of the mitosis that is actually called as karyokinesis. Okay. Then at the end, like once everything is sorted, right, and the cell is initially ready to divide. So during division, what happens? First thing is the chromosomes are going to divide, right? So that means daughter chromosomes are going to separate. That is karyokinesis, and the entire cells gets separated by the formation of furrow. That is the cytokinesis. Have no like what exactly the karyokinesis and cytokinesis is. So, since the number of the chromosomes in the parents and the progeny cells are the same, that is 46 number of chromosomes, it is also called as equational division. Say, like it has only 46 chromosomes, then obviously in daughter cells also you're going to see the 46 number of chromosomes itself. Through the mitosis have been divided into four stages of the nuclear division, and here where the karyokinesis will occur. So prophase, metaphase, anaphase, and telophase. Coming to the prophase, which is actually the first stage of the karyokinesis of the mitosis, followed by the S and G2 phases of the interphase. So what happens here is in the S and G2 phase of new DNA molecules form. So S phase is something DNA like the DNA got replication, you will see, synthesize, you will see, right? So, what happens? The prophase is marked by the initiation of, you can see the thread like structures of the DNA here. They gets condensed to form like this, okay? So, condensation of the chromatin will occurs. So, now uh, these thread-like structures will get condensed. That is, the chromatin gets condensed. Okay, so this is a keynote difference, early prophase and late prophase, where you can actually observe it very clearly, right? The centromer, which has undergone duplication during S phase of interphase, now begins to move to the opposite pole of the cell. So what happens here? The chromosomal material condenses to form a compact mitotic chromosomes, that's for short. And the chromosomes are seen to be composed of two chromatides attaching at the centromere. So if it is a centromere, so what we are going to observe is two chromatins, okay, they are been attached to the centromere. That is initially what we are going to observe like this. So the centrosomes, which have undergone duplication during the interphase, begins to move towards the opposite pole of the cell, and each centrosomes radiates out the microtubules called as aster. So what happens is in this phase, the centromere, which was been divided, right? So it is going to radiate out something, a thread-like structure, which is actually called as asters. And the two asters together with spindle fibers. So what is happening here? So these are the spindle fibers. Okay. So it is going to radiate the spindle fibers, and the cell at the end of the prophase, when viewed under the microscope, you are not going to find any Golgi complex, then endoplasmic reticulum, nucleus. Also, you are not going to find, and nuclear envelope will also be absent. After prophase is followed by the metaphase, by the stage the condensation of chromosomes is completed. Then the morphology of the chromosomes is most visible, easily visible. Then the metaphase chromosome is made up of two sister chromatides. So one and two sister chromatides, which are held together by this is actually what? The centromere. They are being held together by the centromere. So you're going to see the two sister chromatides, and which is held by centromere. A small disc-shaped structure at the surface of the centromere are actually called as kinetochore. So whatever the disc-like structure that we actually see here is actually called as what? Kinetochore. Okay. So yeah. So these structures serves site of attachment of the spindle fibers. So whatever the spindle fibers are radiated, so to the structure, okay, the spindle fibers are attached to the chromosomes. And the plane of alignment of the chromosomes at the metaphase is referred to as the metaphase plate. That means after a certain movement, what happens? The chromosomes will arrange in a very plane position. So they come to the metacentric position and arrange as a plane-like structure, which is actually that's why we normally call it as metaphase. They're arranged equally at one metacentric phase. So spindle fibers which has been attached to this kinetochore of the chromosomes, and the chromosomes are moved to the spindle e- equator and gets aligned along the metaphase plate through the spindle fiber to both the poles. So after metaphase is followed by anaphase. So each chromosome, what happens? They which were actually attached in the metaphase plates, right? So they get split simultaneously into two daughter chromatides. So here they got split into two daughter chromatides, and they are actually referred to as the daughter chromosomes of the future daughter nuclei, being that emerge towards the two opposite poles. That means they're actually moving to the two opposite poles here. As each chromosome moves away from the equatorial plate, so if this is the equatorial plate, and the chromosomes will, it will start moving opposite to the equatorial plate, the centromer of each chromosomes remains directed towards the pole, and hence at the leading edge with the arms of the chromosomes trailing behind. That means these are the arms, leading edge. So this could be a leading edge, and this is the arms. So leading edge will always be moving forward, where arms of the edge, arms, will be taking its lead. That means it is going to follow the lead. Then the centromere split and the chromatides separates. Then the chromatides moves to the opposite pole. This is the keynote that happens in the anaphase. After anaphase is followed by the telophase, which is actually the last phase of the M phase. So at the beginning of the final stage of the karyokinesis, what happens? The telophase, the chromosomes that have reached that respective folds, they gets decondensed. That means you are able to see the chromosomes now. Now they are going to decondense, lose their individuality, and you are going to see no longer, you are not going to see the visibility of the chromosomes, and each set of the chromatide materials tend to collect to each other and pool and all. then the chromosome cluster at opposite spindle poles, and their identity is completely lost. Again, they are going to become thread-like structure. So nucleus appears, nuclear membrane reappears, okay, and you are going to see what? Now, in one cell, you are going to see two daughter nuclei you are going to observe. Then you will also see the Golgi complex, endoplasmic reticulum, nucleus. So they all get reappear in the telophase. So now the telophase is ready, and equally what happens? The all the organelles have been divided, nucleus have been divided, then the chromosomes have been divided, DNA, everything has been separated. Now the cell has to divide. So if this is a cell, parental cell has to divide into two daughter cell. So that is called as um, cytokinesis. So karyokinesis happened where the DNA got divided, right, into equally, splitted into equally. So we got to know how DNA got divided, how the genetic materials have been separated, and all through the phases of M phase. Then the last where the cell has to divide is called as cytokinesis. That is the side, uh, you know, the outermost. So after the mitosis is accomplished, not only the segregation of the duplicated chromosomes into daughter nuclei, which is actually through the karyokinesis, but the cell itself has to divide into two equal daughter cells by the separation of what? Cytoplasm. So that is called as cytokinesis. So in animal cells, it has been achieved by the appearance of, this is actually called as furrow. So whatever that you are observing here, right, it is furrow in the plasma membrane, and it gets deepen, and ultimately what happens? The cytoplasm gets separated and the cell gets divided. In plants, what happens now? Instead of furrow formation, what happens? So that is outside the cell, what happens? It meets inside. That is, in the plant cell, the wall formation starts in the center of the cell and goes outwards. Okay. So if it is a cell which is ready to divide, so wall formation will occur here, then it is going to grow outside. Wall. The formation of the new cell wall begins with the formation of simple precursion cell, that is the cell plate, which represents the middle lamella between the walls of two adjacent cells. In the plant, I'm talking. Okay. So at the end, what happens? The cell eventually, the cytoplasm also gets divided, well, leads to the formation of two daughter cells. And they also get matured, becomes parental cells. Again, division will occur. So this was all about what? The very first initiation of cell cycle, that is interphase and M phase. Okay. So there is another phase which is actually called as meiosis phase, which is very important phase, and it has several processes with respect to the meiosis phase. So which I'm going to cover again in the next video. But for now, this video, I have covered what is two very important phases in the cell cycle, that is the interphase and M phase. So regarding this, if you feel any doubt, you can reach me out to the message section. And yes, very important thing is don't forget to like, subscribe the channel, and comment, and also share with your friends. Thank you.